Thank you. I'm a little bit awed to be in front of this audience. And as you saw maybe in the program, my title was Plus ça change, plus c'est la même chose. And the subtitle was 40 years of biocuration. And then I made a computation. And so I needed to update, and that it's almost 45 years that I've been doing this type of work. And in fact, when I asked ChatGPT to make a small picture with Swissprot and Celosaurus, I didn't ask the system to add a dinosaur, but it did. So I think that, I mean, the message is clear. I mean, I'm a dinosaur in this field. Now, I mean, just maybe in one slide, I mean, my parcours, story into the world of biocuration. Well, it started in the 1980s when I was a very young student. I wasn't yet a bachelor in biochemistry. And I was writing a software package which became PCGene, and then already typing in protein sequence and organizing them and putting them into the computers. So that was the first approach to biocuration. But really, it was in 1986, which really, I would say, the start of my career as a biocurator by starting Swissprot. And when I started, I never imagined that I would be involved in almost 200 releases of the database for the next 20 and more than 23 or 24 years. And also something which is quite important for me is that I had this pleasure to work with almost 200, we called them annotators, now they're called biocurators, across all of those years. So that was really rewarding to work with so many, people which were, passionate about biocurating proteins and building Swissprot. It started small. I started alone, and there was another person. I remember writing with French scientist Jean-Michel Claverie a grant proposal which was rejected because we were asking too much. We were asking for one person and one computer. And in fact, we think that with one person, we'd be able to annotate all of the proteins that existed. Okay, it was too much to ask for one person, but of course, it took much longer than, a grant period to do it. And it led to the buildup of what became now UniProt. Then, of course, there was, ProCyte and enzyme. And in 2010, I left the Swissprot group to build a new group, which the basis was to do both lab experiment and biocuration around human protein. And that led to a resource, NextProt, which unfortunately, due to lack of funding, and I will come back to funding, this will be a big part of my talk, is dead now. So NextProt doesn't exist anymore. While of course, Swissprot under UniProt, KB Swissprot, ProCyte, and enzyme still exist. And in the last 10 years, I've switched completely from protein to cell line. It was an accident. I didn't want to create a cell line database. I just wanted to use one, and there wasn't one. So basically, it led to Celosaurus. Now, if I now, go back to biocuration as such. In 2009, there was the first meeting of the International Biocuration Society in Munich. And I wrote, in my talk, what people have called Bairoch's lament, is that at that time, I was depressed to think that we're spending millions in grants to basically perform experiment, which is great, but then we have to spend money to extract information from papers and basically spend millions trying to get what was obtained by those experiments. Now, this lament is still, I think, valid. A lot of things change in the last 20, 30 years, but in terms of publishing, we're still publishing papers as they were, 20 or 30 years ago. There are more journals, there are more, the whole ecosystem has changed, but basically people are still writing papers, which is nice, but are hiding, in fact, information in table, in figures, or basically in supplemental figures. And the work is still, to try to extract, this knowledge from those papers, which is a bit, disconcerting, because you could have thought that, I was hopeful 10 or 15 years ago that people would start writing paper with the help of tools that would, at the same time as the paper is written, write, computer, readable version of it. Anyway, things have changed in the last year, in a number of years. And of course, two things for me strike me have changed. One is so obvious, and all of you which are in your audience are much younger than me, so for you it's obvious. All of the papers are available online now. It used not to be the case, and people, biocuration meant going to the library, photocopying thousands of pages, and then reading those, on your desk, while now everything is on, of course, available. I just did some statistic out of almost 30,000 publications which are cited in Celosaurus. All but 1,200 were available as PDF somewhere, and only 1,200 had to be scanned or ordered from the libraries and so on. And only 30 could not be obtained. There were theses, there were obscure journals which have disappeared. So it's, everything is available. Of course, one thing which helped, and I should mention, is even so that Switzerland is one of the rich countries and has a good library, subscription system to major, publishers, not every paper can be obtained free of charge. And of course, if you have to pay, if you're building a database and you have to order, articles from publisher, you would basically run out of money from your grants, after 1 week. And one site helped getting this free, to allow biocuration. Now, the big, I would say, change in biocuration in the last 30 years are ontology, and I would call supporting resource. Not only ontology, but everything which allows now bioresource and curated bioresource to have a stable, foundation, and to speak with, basically the same vocabularies across different resources. Just as an example, if I take Celosaurus, we're using, a number of ontology, Uberon, for tissue and organs, vertebrate breed ontology, ordo, and for rare disease, and things like the NCBI taxonomy, Flybase for Drosophila gene, and so on. You see the list here. And this is just an example. Each of you working, if people in the audience are working on, some, bioresource, they will have their own, set of ontology and supporting resource they use. And that made basically, I would say, a big change in the world of biocuration. Now, I had to train a lot of biocurators over the years. And at one period, I was pondering what is so important for one to become a good biocurator. And of course, you need a broad knowledge of the field. You should not be too specialized. You need to have quite a broad knowledge. That's more or less easy to obtain people with this. But I think one important thing is this never-ending astonishment when learning about new discovery. The best biocurators are excited when they look at a paper and say, hey, they found, the reasons that this gene is doing this, that this protein is acting on this. And that's basically help really to curate. If you're excited about what's happening in the field you're curating, then chances are you're going to represent it, in the best way possible. Now, also one thing which is important is not to be discouraged, to be swimming against the current. You never get your ink tray going smaller. You start with some things to curate, and 2 years later, you have twice more, and it goes on and goes on. You're never finished. And not only are you never finished, but you get more work as time goes by. So the number of things to curate always is increasing, and one should not be discouraged by it. You need, of course, a synthesizing mind, but that's more or less goes with the work. And I would say also the best biocurators have an uncanny ability to spot errors. I remember when we trained biocurators in Swissprot, we told them, if you read a paper and you don't find an error, you didn't reads the paper well enough. You have to find errors. Errors are everywhere. It's normal. And basically, a biocurator needs to spot them very quickly in the papers, but also in what they produce in their database, because, of course, they will also make errors. And you can have programs to help you check things, check for consistency, but you still need this ability to spot errors. And the best biocurators who can really spot errors very quickly. I think you need a samurai drive toward perfection. You need to want to be perfect. Of course, you never achieve perfection, but you need this. And I would say almost an autistic attention to details. I think that's basically what people, need to be, the best biocurator. They need to be an autistic samurai for biocuration. Now, in terms of biocuration and resources which are biocurated, there's always been a problem of funding. The word plus ça change, plus c'est la même chose means basically to acknowledge the fundamental immutability of human nature, and in this case of funding problems, and I will come back to it. If you look, this is a paper from 1986 when, Swissport was in trouble. This was one of the first, I would say, article in a scientific newspaper speaking about problems for a biological database in terms of funding. But of course, it continued, and every 4 or 5 years, you get articles about those problems. Of course, things are now worse. I'm not going to go in this. And biocuration is only a tiny part of the, I would say, of this huge problem and huge issue, which is, now rolling the world due to what's happening in the US. But of course, biocuration is also impacted by what's happening in the US. Now, if you look at what funders, funding agencies think of biocuration, I think it went every 10 years, it changed a bit. In the 80s, they didn't understand. Why do you need database? There are journals. Scientists read journals, read articles. You don't need a database. It was simple. You don't need to do this, so we don't need to fund it. 1990s were, I think, the golden age of funding. Suddenly they thought, yeah, maybe there's too much data that people can read them in article. We need to build database. So we'll fund you for 3 or 4 years, and then you get everything done, and then we can move on. So there was this idea, yes, let's fund Swissport, let's fund other things, and once this grant is finished, it'd be good. Then, of course, 10 years later, it was, well, you're doing this manually. You have people working on it. Use text mining, and this can be done automatically. And then 10 years later, well, you're doing this with curators you're paying for. Ask the community to curate. They will curate, they will do the work, and you basically take everything from the community and put it in a database. And now, of course, AI and deep learning will replace manual curation. So every 10 years, there's been those criticism of biocuration. Users also, do not often recognize what biocuration is. If you go back also in the 80s, for most, I would say, biologists, somebody wanting to biocurate was somebody it was a failed scientist, basically. We wanted to play around with computers instead of doing experiments. And a PI didn't really understand why you needed a database. They knew everything they need to know on their field. Then, again, the 90s was this golden age where everything was, oh, yes, that's great what you're doing. And basically, also PIs thought, yes, that's good, get the funding, and hire a junior scientist to finish the job. 2000, then people started to gripe. You haven't finished it annotating all human protein, but the human genome was out 2 years ago. What are you doing? Basically, it's only a 20,000 protein to annotate. Get on with it. And then, oh, you mean there are people doing this? It's a job. It's not done, automatically. And of course, now you have people saying, well, I don't need real database. I ask ChatGPT about it, and that's, or Gemini, or whatever is your, preferred tool. Now, yes, that's great. But then that's the elephant in the room. I mean, basically, biocuration is really essential to provide the structured and high quality data which is needed by those large, models, language models. And those building those models are aware of it, of course. They're expert in it and they know they need good quality data to train, but funding agency and people don't realize this. And it's becoming really, under-recognized, by funders and by many users, which thinks that you don't need biocuration anymore. ChatGPT is going to do it for you. Now, ask ChatGPT, why is there not enough funding for biodata resource? And for this type of thing, it's great because he answered with 7 different reasons, and all of them are really spot on. Databases are often seen as infrastructure, not research. Completely true. And of course, if it's not research, it should not be funded by research agency. There is a lack of dedicated long-term funding models, and that's true. And we were lucky in Switzerland to be able, thanks to the crisis of Swissprot in 96, to create in 98 the Swiss Institute of Bioinformatics. And that led to stable funding for Swissprot, for other resources. Unfortunately, not enough, because the number of resources has grown in the last 25 years, and the funding has stayed stable. But still, I think we're very lucky in Switzerland because we have a long-term funding model, but it's still not sure that this model can continue because it's funded according to an obscure article of law in the Swiss ecosystem of funding, which people are criticizing. So we don't know what's going to happen to this stable funding. But still, I would say Switzerland has this model, but most countries do not have a dedicated long-term funding models for biocuration and for bioresource. Invisible usage and other citation, yes. A lot of people are using database, but do not cite them or do not mention them in paper. Misalignment with academic incentive, that we say is less true, because, but still, people wanting to do biocuration are not going to be, seen in a very good light by, people hiring, professors or researchers, but that's, less true now than it was before, but still true. Explosion of biological data, yes, we all know about that. Global use, local funding, that's quite important, it's true. If you look at most of the bioresource, their funding comes from 1 or 2 sources. And if there is a problem with this source, then you're in trouble. And basically, the use of those resources is global. And it's also difficult to get local funding because, let's say, when we go to Switzerland and we go to the politician and we say, look at what the ECCB is doing, what it's doing with those databases and so on. They say, yeah, but how much percentage is used in Switzerland? And we have to say, well, a tiny bit. It's not the biggest user by far, of course. And they say, but why do Switzerland has to pay for a resource which is used by every other countries? And you have to explain, yes, but it's useful, it gets, recognition and so on, but still, the question remains, it's globally used, but funding is local. And last but not least, if it works, why fund it, paradox? So I think that ChatGPT had summarized quite nicely the 7, reasons. So one thing is, what is currently done to fund biodata resource? So it's 2 big initiatives. Elixir in Europe, its mission statement is to build a sustainable European infrastructure for biological information. And recently, we have the Global Biodata Coalition, which convenes the world research funders, so it's a club of funders, to exchange knowledge and share strategy for supporting biodata resource. Now, each have made, list of resources that they think are fundamental. They basically ask resource to submit, basically, a description of the resource, of the usage, and ask, experts to judge it. And every year they came out with Elixir. It's every 2 years. The Global Core Biodata Resource did it twice. They came out with list of essential resource. So Elixir Core Data Resource and the Global Core Biodata Resource. That's nice. It allows database which are one of those resources to decorate their own page with nice logo. So how much money do we get from them? Nothing, nada. Basically, discussion are underway. Some, little grants are provided by Elixir to do some community effort and so on, but not, no progress has been made really to find, any funding for biodata resource. And with the situation like it is in US, everything is now worse. So what is the solution? I know I'm not going to make a lot of friends here, but I think that if you look at different, community, look at the chemist. One of the biggest databases is Chemical Abstract. Look at legal, the domain. So have LexisNexis. Now, if you look at those efforts, CAS Chemical Abstract has 4,300 employees. LexisNexis has almost 12,000. Now, let's forget about the lawyers. They're rich, they have money, and so on. Law firms. But Chemical Abstract, you have 4,300 people abstracting information on chemical. That's more than all of the number of biocurators in the world doing all of the biocuration. Just CAS has more curators than all of our life science. But, what's important for us is a FAIR principle. And I totally agree with it. And in fact, Swissprot from the very beginning was fundable, accessible, interoperable, and reusable. We didn't know that it was going to be called FAIR. And you have those FAIR principle. But look, the F in FAIR does not mean free. And people think that FAIR means that things have to be free. No, they have to be fundable, accessible, interoperable, and reusable, not free. I know that, some people would disagree with me there. In fact, when the crisis of Swissprot happened in 96, 10 years after creation, we didn't have any funding. We basically created a totally asymmetric model where academic user, non-profit user would continue to use the database free of charge, and company had to pay. And everything was done based on trust. So no login, no firewall. You were an academic, you didn't see any difference except that each entry in Swissprot had a small square, 5-line saying, if you're academic, just go on and do whatever you want. If you're not, you need to have a license and apply for this license. This worked fine. And basically Swissprot both at that time in Geneva and at EBI in UK got basically a lot of money through those, license from industry. And unfortunately at the time, and it's quite paradoxical, with the situation now in US where, everything seems to be that public money should not be spent and it should be, done differently. At the time, NIH and other agencies didn't like it. They say you basically, doing something which is asymmetric, so you are, asking industry to pay and not academic user, but you should have everyone, basically be on the same level, and in this case, be free. So basically pressure was applied, a lot of pressures that we stop having license of Swissprot, and it led to an NIH grant. And when I returned from University of Geneva 2 years ago, I said that basically that was probably the biggest, mistake of my career. And in fact, since then, it has been very hard to get money from Swissprot and for any other resource. Now, a lot of resources have also already started asking for, license fee, TARE for plants. You have different resources which do this. But I think what's Cosmic, for example, for somatic mutation. But I think what's very important is to try to find a model where it's totally seamless for academic users and for nonprofits, so that there is no login, no barrier in usage, and that basically only the people which can afford it, the companies, the pharma company which make millions out of those efforts, pay for this. I will stop at that part and just have one slide which is more scientific, which is on cellulose, because that's my passion in the last 10 years. So in one slide, what is cellulose? A number of you know it, but not so many, because in fact, the world of cell line is quite different from the world of gene, protein, and so on, of which you're all aware of, and most bioinformaticians are working on sequence or sequence-related aspect, basically, and not really on cell line. Now, cell line are quite interesting because, in fact, I didn't know anything about cell line, I have to be honest. I mean, except they existed and people were using them to do experiments. But I had no clue about the intricacy of the world of cell line and the mess it was in terms of organization of data on those cell lines. Because, in fact, the only people really collecting data were cell line collection, and they were only interested in creating or presenting information on the cell lines they were distributing, but not on other cell lines. And a lot of cell lines are not distributed by any collection, but are basically given by one lab to another lab, and then maybe trouble happens because some of them get contaminated and so on. So it's a very interesting world. I won't have time to discuss it. I could discuss it for 2 hours, but it's not, I don't have time to do this. So what is Cellosaurus? It's a knowledge resource on cell line. It's almost 170,000 cell line already. Yeah, when I started also, I thought it would be a list of 1,000 cell line. Again, I was surprised to see how many cell line exist. Some of them have disappeared, but they're still important to, document because they were used in experiments for a number of years, and then people stopped using them and so on. So almost soon 200,000 cell line, and a lot of different type of cell line. Some are naturally immortal, like pluripotent stem cells or finite cell lines. Some are immortalized by virus, by chemical. I'm not going to go into it, but basically, Cellosaurus covers all of those different type of cell line, vertebrate, invertebrate, and since a number of 2 or 3 years ago, also plant cell line. Now, the number of information items in Cellosaurus has grown. It used to be only the name of the cell line and a few cross-reference to cell collection, but now there's almost 50 different type of information items, many literature reference. I spoke about that at the beginning of the talk, saying there's almost 30,000 reference, and cross-reference to 115 resources, so links to cell line collection, to database, to ontologies, and so on. I already mentioned different ontologies which Cellosaurus use. It's, of course, available on the web, on the XPASI server, with the name cellosaurus.org, available by FTP, also in different format, and we have an API, which also you can download in JSON. And now, since a month ago, we're really happy to have a Sparkle endpoint. And basically, you can access now Cellosaurus through the Sparkle endpoints, download it in TTL format. Basically, it can now be part of the semantic web. It's collaborating with a lot of different entity. You don't know them really because they're basically specific to cell line, except maybe the Resource Identification Initiative, which is quite great because it allowed people, it's asking people and journals to have people citing resources they use in experiments. Antibodies is a big chunk of those resources, but cell line also, plasmid and others, and to have a unique resource identifier for all of those experimental resources. And it's recognized, I said it already, as an Elixir core data resource, a global core biodata resource, and so on. So that's in one slide, Cellosaurus. With this, I will basically end early to let time for questions, but first by thanking all of those people. Some of them got multiplier, like you see Alain times 3, and so on. That's all the names, the first names of the biocurators with whom I had the chance to interact either in Geneva or at EBI or elsewhere. And that, I think, is the most important slide. Those people are the community of biocurators which helped build SwissProt, and ProCite and Enzyme and NextProt and now Cellosaurus. And with this, I will end, and thank you for your attention.
We have plenty of time for questions.
But we don't see anything.
Okay. Go ahead.
got a ball
Bonjour
here.
Amos, put ici.
Amos, it's a fantastic presentation. And what you
Oh, there.
hinted at was something I'd love to know more. How are you approaching the problem of encouraging the generators and the users of the cell lines in Cellosaurus to provide adequate metadata so that you can perform the curation you're describing?
Yeah, I mean, metadata for cell line is even more complex in some aspects than metadata for, proteins and genes. I'm not sure if part of it is problem is metadata, but also I would say data which allows to see if a cell line is really what it is, which means information on what we call its STR profile, its short tandem repeat profile for human cell line, which allows to know if a cell line is contaminated or not by basically doing what people do when they do a paternity test. We check those STR profile against a database. That's done in Cellosaurus, and to see if a cell line is contaminated. And this is getting better because journals are more and more aware of the problem of contamination and asking people to submit this information or at least to authenticate cell line. Now, for the rest of the metadata, it's a big battle, as you know, for every field to get journals to insist on getting enough data. We're already happy if people cite the cell line with its accession, Cellosaurus accession, which is a resource identifier, so that we can know which cell lines they think they have used. Of course, I say think they have used because one of the big problems with cell line is people getting cell line from the lab next door, which is nice and fine. We did it also when we were doing experiment. We asked a lab, do you have, a vial of MCF7 cell line and we can share with like this, we don't need to buy it from ATCC. Oh yeah, don't worry. And of course, as cell line can grow, you just, take a vial and you use it. Problem is, if that vial is contaminated, you're doing work on contaminated cell line. And that's, I would say, the biggest issue in the world of cell line. Contamination by cancer cell line like HeLa are a huge problem. I don't know if I answered your question or. Okay.
Thank you so much, Amos, for your inspiring talk. I have a question related to the database scarcity, because there are so many databases and the information is scattered here and there. For genomes, there is different database. For pathways, there is CAG database. For BioSeq, for curation of genome scale metabolic model. So my question is since I am working on genome scale metabolic model and genomic analysis, pan-genome analysis, and I always feel this problem for getting all the data from different resources. For genome scale metabolic model, you need different kind of data to enrich your model. So what are your views to have one particular database having all the informations? Because you are an expert in biocuration and you have established this field, so I thought you are the right person to ask this question. And how much resources we will need to host such a database?
I don't think there will ever be a unique database with everything. What's getting forwards slowly and slowly is that you can interoperably make those interoperable, thanks, I would say, a lot to ontologies and so supporting resource. If everybody, for example, using GNAME is using the AGNC, accession number for the GNAME, we know we're speaking of the same gene. People, when you used to put database together to do work, most of the time you were spending days and months trying to align different information, trying to see this is really the same gene or same protein as this other one. And I think it's not perfect yet, but I think that this has changed. And thanks to things like, RDF and Sparkle endpoints, once you have those common vocabularies, you can do federated queries across database. And of course, it's not trivial, and it will sometimes fail, and it will take sometimes huge amount of time to get those. But you can do much more than you used to do without having to put everything into one big silo. It's not perfect. It will go forward slowly, and ontology and techniques like RDF and Sparkle are the way to help going in this direction, not building a huge silo which nobody has the money or so, or even know to build this, and it would be basically unmaintainable. Better to have specialized resource doing very good curation on what they're expert in, but making sure that they can speak to each other.
Thank you so much. I have one more follow-up question, if it is allowed.
Yeah, go ahead.
Okay. My next question is like, because you said like databases are linked, but the problem is like in each database, nomenclature is different. So for one particular gene, the information will be different, like interpretability. And for if you classify this as an enzyme or any other thing, the information is completely different. So it is always a problem to integrate all information in your particular model. So how do you address this? Like, is there a kind of like universal nomenclature, and what are your suggestion on it? Like,
But again, I think it's ontology are part of the discussion, and also not only ontologies like Go or Uberon that we use, but also the ontology of the resource you're creating. When you create an RDF model of your resource, you need to also describe it as an ontology and use as much as possible ontological terms which are shared by all of effort. To give a simple example, every resource is using reference to publication. Now, there are some standards in way to represent reference in the semantic world. And if you use those standards, you can basically exchange reference between one resource to another. If you create your own silo of RDF reference, then of course you have this problem that even showing that it's the same reference will be difficult. But it's part of the work of people building database to make sure that they use ontology and that they describe their resource as an ontology also.
thank you so much. That was wonderful.
So we'll alternate microphones. Now, number one.
Hi, bonjour Amos, c'est Francis Ouellette de Montréal.
I don't see where you are, but I hear you. Oh yeah, sorry.
So thank you very much for your talk. It's great. It's always great to especially for the younger people here, to see the historical perspective on a great field of science. I was at NIH in '96, and as you recall, and yes, NIH was pretty pissed back then. And so the main reason was that our affair wasn't available anymore. So it wasn't reusable. Swissprot, it appeared to us at the time. That was one of the big concerns. And I'd like to propose to you that you today proposed two alternating sort of funding models, one where funders pay everything and the other one where the industry pays for it. I would argue that industry is not all big pharma. Industry could be a small biotech of 5 people, and they don't necessarily have the money to use resources, and they become disadvantaged. So I think there's been a lot of talk of Trumpism and stuff like that at this meeting, and there will be more. And I think it's important to think about other models, not just those two. I think we need to think about 3 or 4 other models before we can sort of change the way we do things. And I regret how your biggest mistake of your life was to get money from NIH, but that's not a bad one, actually, if you think about it. But I think we have to, it's true that we should not depend on one resource. And so I applaud that. Thank you.
No, I completely agree. We should not depend on one source. So public funding should try to hopefully stay and allow academics to be basically, it would not be free for everyone. Everyone would pay, but academics would not pay directly, but pay through public funding. And when I say pharma, I didn't mean only big pharma. And if you remember, Swissprot had a model where you had 4 or 5 different categories of company, which is now, basically, so what people call micro enterprise. Small, medium, large, and huge company. And of course, micro company startup would only pay something like $1,000 per year to get Swissprot. I disagree by saying that it could not be available, Swissprot. If you were academic, you could FTP, you could use it. You didn't have anything to do. It was, if you were a company, you had to do it. So availability, it could, you could not completely integrate it into NCBI. Yeah, because of course, reusing it and integrating completely, I agree with you. That's one of the problems, is if somebody integrate a resource versus license and put it free to the public, then of course, then you have this problem. Yeah, I mean, any type of hybrid model needs to be thought out, but in fact, I think we really don't have a choice. We cannot rely only on public money anymore. I mean, it didn't work for a number of years. Databases are underfunded for so many years, and it's getting worse. And now, with what's happening in US, it's even, more catastrophic. And so, any model that people can think of, I am a buyer for that model.
Back to number 2.
So, well, my question was pretty much the same as the funding question that has been raised before, but I think that probably it's important to raise awareness of for the public funding of the databases. Because there are many databases, some of them maybe not of primary interest to the industry, but for the global research community. Mm-hmm. But I think also, in a way, the databases are collecting the research of the community, and many people are putting their data or their knowledge into the databases. It's not just that it's nice to have a database, but it's also where the knowledge is safeguarded. Mm-hmm. So I think that in the same way that the public institutions is funding the generation of knowledge, it must be demanded that also the preservation of knowledge is funded in the same way. So now maybe it's the moment in which, with the US changes, that there is a stronger voice from Europe to ask and to demand that there is a serious investment of taking care of all the knowledge that has been generated by the whole community.
Yeah, I agree with you. But so unfortunately, for example, if I look at situation which I know well, which is Switzerland, which is only a tiny part, we have this big effort in Switzerland that every data which is produced by people getting a Swiss grant from a Swiss National Science Foundation needs to be stored and put in database and be reusable and so on. But at the same time, this is basically telling scientists, do the job, but it's not providing money to do it. So, scientists are grumbling about it because they say, oh, no, we're forced to do this and we're spending time, which is great that they're doing it. But, once they ask for money to put it on their grant, so it's not really a mechanism you can now ask for money for computing time. But so for example, Swiss National Fund accepts that people say, I have this research project, I will need so many hours of CPU, please add in my grant this amount of money. But when we asked at the Swiss National Fund, would there be a way of doing this for biocuration? The answer was, oh, that's a nice idea, let's talk about it, but nothing happened. And but it would be a solution also that grants would include money for biocuration, either by the scientists themselves or to go to the database to help them doing it.
Thank you.
It would be wonderful.
Number 1.
So, coming from Australia, I'd like to note that we also had something like NCBI, which suddenly stopped getting funded a very long time ago. So funding from the government or public agencies is not something anyone should rely on. And I have 2 solutions which I think could be used. One of them is open access kind of model where everybody is now paying for their publications. So could institutions sign up payment for submission to databases? Number 2, we had iPod all those years ago when Steve Jobs suggested that people pay a minuscule sum for each music track they downloaded. So could we have a single access fee for each entry? Provide the search, but when they click on the link, they will have to pay a particular amount, or they can have their own account with some credit on it. So that way the user is able to pay small bits of their research funding, academic or commercial or whoever, and then, you start collecting money like crowdsourcing. So these are my 2 solutions, and we can talk about it later. Thank you.
In principle, I agree with you. It would be wonderful to have a system of micropayment and also micropayment if the scientists getting back money if they help create information. Technically, I mean, it's not a technical issue, it's more an issue of getting this organized worldwide, because it cannot start only in one country and so on, and getting people to do this in a way. And I think that if there is not a huge organization somewhere coming in with idea, or a billionaire or someone, saying, let's do this, then we can talk about it. But nothing will happen, because I remember discussing this already at an ISMB meeting in Fortaleza in '96 in 2006 for the 20 year of Swissprot, and we were speaking of getting the community and getting, micropayment maybe, but how do you organize those micropayments, and how you organize an organization which will do those micropayments? Will not do, but will basically facilitate this and make a system which is fair for everyone. But I agree with you, it will be wonderful.
Hello, thank you so much for this really wonderful discussion. I have another micro solution to offer, and that is that everybody in the room can do, is when you're using a resource, whether you are thinking about it or not, make sure you cite it in the references in your text. So not just like a web link, www, or the persistent identifier, but when you use them, put it in your reference link in your text. So for Flybase, for example, which a lot of us use, love, know, had only 129 citations in 2024. So you need to actually call out the database and the reference that you are using. If you are using an ontology, anybody here using an ontology, have you put the ontology in your reference list? When you do that, you give impact to the resource, and then you give people arguments they can use for their funder. Look, my thing was cited 30,000 times last year. So not just usage, citations. Thank you.
I totally agree with you, and that's why, that was one of the things ChatGPT said, under citation. And that's why, 10 years ago, there was, I think it was Phil Bourne, a student trying to build something with Microsoft where there was an app which would do this type of thing. So that when people would start writing an article saying, and I've used, the Go ontology and extracted from Swissprot, it would add those type of things. But we still haven't that. And of course, scientists said it's not that they don't want to cite, and journal generally tells them to cite things. But we're not making them easy to do so, because let's say it's only use 10 or 15 resources, which happens quite a lot now with papers. They have to go to each resource and look. Each resource will have a page saying how to cite us, but it will be hidden somewhere, sometime on the top page, sometime in a different page, and so on. So basically, we're not making the work easy to people to cite things. And I think part of it could be a software solution where, something is written, or you basically, write your article and ask ChatGPT to add reference to all of the resources. That would be maybe the simplest way now. You ask basically, I've used those 15 resources, find me the best citation for it. Because it's often not so easy to cite resource due to this fact that you have to go everywhere. And and people are more, when they write a paper, it's easier for them to cite another paper because they use that paper than to cite the paper of the database. And yes, it should be done.
Given everything you found out from funding issues with Swissprot, how did you change your approach with Celosaurus funding and getting money for that project?
Well, we're thinking about going with this idea of a license. We're discussing it. It's not yet decided. So funding for Celosaurus, it's easy to explain now. I'm retired from university, so I work pro bono, basically full-time on it. So funding is my retirement salary, and one salary from SIB, the Swiss Institute of Bioinformatics, according to this funding scheme I told you about, which Switzerland is funding resource, and each resource applies at the SIB to get resource, and I got one position, which I'm really happy about it because it allows to have a developer, software developer, which wrote the Sparkle endpoints, the API, and so on. But curation, basically, of Celosaurus is not paid at all. I tried to see where I could apply, but curating cell line is even worse in terms of getting fundings than protein and gene, where you have a lot of different call. I'm not saying it's easy. I know how difficult it is to get funding for protein. Next part, we failed. But for cell line, it's even worse. So I think the only solution would be to ask pharmas which are using cell line and which are spending millions, on cell lines and building this to pay for, these efforts.
Thank you.
Amos, I was struck by your description of the role of biocurators. You said one part was detecting errors. So in the context of the reproducibility crisis, this infamous claim that 50% of research findings are probably wrong, I think that was John Ioannidis. The resources like Celosaurus or the ones we operate at the ISMB, are we, if we're using research literature for our databases, are we perpetuating those errors, or are we a corrective factor against them?
Well, I think a big part of the work of biocuration is correcting errors. And that's, especially I would say biocuration, I often told biocurators, when you read an abstract, think of the abstract as an advertisement. It's not reality of what there is. Find what was really done in the figures, in supplemental, and try to represent what was done really, correctly. And it's, I'm not saying database will be perfect and will not propagate errors. For sure, if somebody found something wrong and it seems, good quality from reading the paper, it will be represented as it is in a paper in a database. But I think database have done quite a lot of work correcting things and also correcting ambiguities and trying to at least clean up some of the errors in literature. So database have a big role in cleaning up data.
Oh, thank you so much for the talk. So I want to ask from the perspective of a high school student, because I myself am one, and it's really interesting listening to the past 40 years of biocuration. So I want to ask about, I want to pivot to maybe the next 40 years, because as a high school student, I know a lot of students are interested, especially with both using and also contributing to these resources. So what is your vision to set up these modules and initiatives to reach out to maybe undergraduates or even high school students to get them engaged in this ecosystem and maybe cultivate the next generation of biocurators? Thank you.
That's a great question. I think organizations like ISCB and others have a big role to play in education. We probably, biocurators and biocuration database are probably not doing enough for outreach. Swisspot, we had the luxury for a while to hire a number of people which were doing communication for the public and trying to get high school students. We had somebody in Swisspot which was going to high school in Switzerland and explaining to them what was a database and so on, and how useful it is. But we're not doing enough about that. And we're not even doing enough, I would say, not only for high school students, but for university students. And so we got a big problem in terms of trying to get the message about biocuration, how useful it is, and maybe to try to explain that to people. And I agree with you, that's quite important, but I don't have a magic solution except to ask everyone to contribute to that, because everyone can do it on their own to try to go in schools, teach in first year students at universities, trying to explain to them what are those databases and how useful it can be, and ask them if at one point they would consider trying to help. Great question.
Hello. So I'm asking a question from the perspective of someone who works on the teaching staff of a medical school in the Global South. So I'm from the University of the Philippines in Manila, and we have a lot of we have a fair number of graduate students, including for like physician scientists. So I think that there is potential to leverage that crowd. I mean, I know that you did present some experiences on crowdsourcing and the results were mixed, but is there any reason to feel confident that we could sort of still leverage that to some extent by maybe somehow aligning the process of engagement and participation with some kind of system by which people could be like rewarded in like very modest ways, modest but significant things that they can eventually accumulate, and it can be on their CV or some equivalent of that. Like, so instead of literally like micropayments in cash, could there be micropayments in terms of equivalent work done? And then maybe on like a grander scale, like, I mean, this sounds like a job for UNESCO, because I mean, this is like World Heritage, right? Like, so I mean, UNESCO is like, I don't know, it's like education, science, and culture, really. So I mean, if we put that all together, and then if the databases could, be elevated to a more central role. I mean, they do play a central role, but as you mentioned, that they're like underrated and underappreciated. But I mean, like, I think the era of textbooks has essentially, passed, or maybe it could be said that it's merged with databases, because even the textbooks now, they're like continuously updated. So the line is blurred, and there are a lot of good resources out there. So do you think we could possibly put those things together?
And I think we tried something with Nextprot. We had a course at the University of Geneva where we teach students to try to find the function of a protein using all different bioinformatic tools and database. Protein, which a function was not known at the time. And they came up with hypotheses, and some of them were well thought out. And we told them, okay, now write with using ontology like Go terms and so on, your hypothesis, and we'll put it in Nextprot with your ORC ID. So they created an ORC ID, and they put this hypothesis, and so basically it's cited in their ORC ID record. And the idea was that maybe if funding agency at one time became a little bit smarter and recognized that, yes, this is not a publication in a journal, but at the time they were students, they did this type of work and they presented hypotheses which were put in a database. So it could be hypothesis, it could be bio-created data. And we thought the ORCID system would allow this, because you can basically put at least on one name, one thing which was done. So the question is, how is this rewarded later in the academic career? And being in a lot of, outside of my university career, I was of course in a lot of those committees to our people. And everyone says, okay, well, let's not judge if they have science and nature. But after 5 minutes of discussion, it came up, oh, but this guy has 2 nature and 1 science, and not at all what they did for the community. And it's tough to get hiring committee to change their mind and see what was done to community is also important when you hire some people, and that a good community player is as important as a good researcher, and it's part of the things that should be awarded. I agree with you.
But do you think there might be some way to, I mean, like, there's a lot of, sort of incentivization. I mean, like, I know that, people get dopamine hits for like uploading a sequence to GeneBank or something like that. Could we somehow up the dopamine hit level for doing that, like for, creating a rigorously crafted database entry or correcting an error or something like that?
Yes, you get a biscuit, maybe.
Yeah, like the other half of the story. Yeah, thank you.
We'll take 2 more questions here, and a last one from online attendees.
Nice road towards addiction. And you. So Amos, this was a great talk, and yeah, I was a PhD student at the time at the EBI in '96 when you had the crisis, and I remember you storming in and out of the EMBL, EBI. This was quite something. Now, over the years, I have seen you training generations of biocurator annotators, and yeah, I remember you mentioning it's about 10 years to train an annotator at least. And we've seen that you're a big fan of ChatGPT, and who is not. Now my question is, yeah, ChatGPT will be terrible as an annotator, and we know this, but it's not going away, and it's a resource we all of us have learned to rely upon. But there's not only ChatGPT, there's Gemini, there is Claude, there is everything. So how does this fit in the future? How do you think the next generation of annotator will have to be trained to harness the power of large language models? And should we develop our own large language model specifically for annotation? Should there be domain-specific LLM? What is your vision of these next 20 years with respect to the interaction with these LLMs?
Well, 15 years, I have no clue, because, I mean, to be honest, things change so fast and so surprisingly. I don't think 5 years ago we would have been able to predict the revolution of those large language models. I'm not the right person to answer. I mean, in the audience, there is Alan Bridge, PI of Swissprot, and I know his group is really doing a lot now trying to harness the power of those large language models to help biocurators by condensing part of articles, by helping finding really, I mean, basically doing better text mining and better representation of the knowledge in the papers. So everyone around, I think, is engaged in, I mean, usage of those models, not by directly typing question and to the system, but using, of course, their API and retraining, I mean, some of the models. And I think this is a revolution, of course. We saw it in all of the field of life science, and biocuration will be, I mean, using it quite a lot. I mean, of course, with all of the safeguard of, you know, making sure that you check and double check, I mean, what is being done, because I mean, the danger of hallucination is, of course, high. There are ways to mitigate this and so on, but still, I mean, the power of those tools is enormous, and biocuration is not different than any other, I would say, endeavor. If there is a useful tool, it will use it. The same was with text mining. When I was saying text mining was the solution, a lot of work was done trying to harness those text mining efforts, to try to use text mining tools, and they're used, but it's not, I would say, panacea. In a way, it helps things get faster, but you still need, at one point, if you're doing manual biocuration, someone looking at the big picture and saying, yes, that looks correct, or no, it doesn't make sense.
Yes,
And,
along the same line, in Oxford University Press is now giving us support at NAR Genomics and Bioinformatics to use dedicated tools for the evaluation of papers. And I find this a very complex process, because we do not know what is the take of these LLMs on the actual knowledge we're looking at.
Yeah.
Last question.
Hi, thank you for this good talk. I'm here.
Oh, yeah, sorry. We don't see anything from the stage. It's a blind
I kind of wanted to kind of continue from the previous question. So these LLMs, like you said, they are the elephant in the room. And as you said, Swissprot and other databases are important as a training source for them. And also they kind of put a layer in the middle, so people not necessarily know that when they ask ChatGPT, it's a Swissprot reference. How do you see that going to the funding side of things? Can you maybe ask them for funding?
Yeah, I think that's a good, I mean, yeah, I thought about it, and other people have said, of course, I mean, groups developing and the software companies developing large language model are using our data, or collectively, or I mean, biocurated data as training set, like they're using all of the literature and everything. And should they pay for it? My, I mean, gut feeling is yes. The question is, we're not alone. Everyone is a little bit angry about that they're using, you know, data everywhere without paying for this, because it's there, it's in Wikipedia, it's in database, everything. We put it free access, and of course, now the views is free to build tools and to ask people to pay for it. And we pay for it and so on. So there is a type of injustice in a way. I don't know how to solve it. I think that companies building large language models are aware that they need to do something, because after a while, if they train the data on data which would have been generated by their own tools or other tools like that, it will stop being useful. So they're deeply aware of this danger, and that they need to be good curated resource, manually curated resource, not only in life science, everywhere where you can basically rely on. I don't have an answer or it's going to work out. I think it's going to work out because every stakeholder has an interest in this big ecosystem of large language model, has an interest of keeping good quality data free of pollution from models themselves. I mean, so I have no clue what it's going to do or it's going to arrive, but it will be a very important development in the next years.
So we will wrap up the session with 2 online questions. One is, could we run research databases like a large torrent network with all resources hosted and shared among its users? And the last question is, what is your advice to young scientists interested in biocuration?
I will answer first the last one, because that's, I mean, I would say go for it. If you're interested in biocuration, do everything to go into this world. If you're passionate, interested in it, then I mean you should go in it. I mean, and try to biocurate what interests you, approach, even if it's not funding, I mean, find a way to persuade someone to allow you to join one of the resources, even if it's partial time, because as it was said before, if you have this type of, dopamine hit, if you're doing something, help database. And some people helping database, submitting information. I have a user of Celosaurus, which sends me every 2-3 weeks. I found a cell line which is not in Celosaurus, it's there, and so on. And I thank him everywhere for that. And it would be great to be able